objectives to establish which solution is behaving as hypotonic, hypertonic, or isotonic, to determine the isotonic point, to establish whether the source is penetrating or non-penetrating.
This experiment will be performed in groups of 4 or 5. For section A1, label the centrifuge tubes as such: hypertonic, hypotonic, and isotonic. Add 5 ml of sodium chloride solution with the corresponding concentrations into each tube. 5 ml of distilled water is added into another fresh tube. Add 50 microliter of blood into each tube by using a micropipette. Step 4. After covering each tube with paraffin, mix the contents well by tipping the tubes with care and leave the tubes at room temperature for 20 minutes. Step 5. Carefully mix the contents again and centrifuge all the tubes for 5 minutes at 2000 rotations per minute. Using the transfer pipettes, carefully transfer all the supernatants into clean test tubes that have been labeled accordingly. Step 7. Determine the percentage of hemolysis in each tube by measuring the absorbency of the supernatants. Set the spectrometer at a wavelength of 540 nanometer and zero it using the blank. Then, measure the absorbency of each supernatant with the same wavelength. Compare the absorbency of the test supernatants to that of the supernatant from the cells treated with distilled water. The last step is to calculate the percentage of hemolysis for the supernatants using the following equation. Percentage of hemolysis equals to the absorbency of supernatant divided by the absorbency of supernatant from the tube treated with distilled water times 100%. For the section A2, the whole experiment is repeated using the different concentration of sodium chloride solutions. Concentration of sodium chloride solution, which are 1%, 0.85%, 0.75%, 0.65%, 0.55%, 0.50%, 0.45%, 0.40%, 0.35%, 0.20%, and 0.1%. Next, add 5 ml of sodium chloride solution with the corresponding concentration into each appropriately labeled tube. For the experiment that have been repeated, the same as the section A1, then we have to plot a graph for the percentage of hemolysis versus the sodium chloride solution. For section B, the whole experiment is repeated using the following solutions. For the tube 1, 300 milliosmol of sucrose. Tube 2, it consists of 300 milliosmol of glucose. Tube 3, it has 300 milliosmol of sodium chloride solution. Next, tube 4 with 300 milliosmol of urea. Tube 5 with 150 milliosmol of urea. And tube 6 with the 600 milliosmol of urea. For the next tube is 300 milliosmol of urea in 300 milliosmol of sodium chloride.
Here is the results for section A1 and A2. Why is that so? We will discuss it now. In this experiment, the distilled water is acting as a positive control with a measured absorbency of 2.296. The assumption made is that 100% hemolysis occurred as there is a huge influx of water into the red blood cells, causing the red blood cells to swell and increases the cell membrane permeability towards the hemoglobin. After centrifuging, hemoglobin escapes through the pores on the plasma membrane. That is why the supernatant is red, resulting in a high value of absorbency. White precipitate can be seen at the bottom, indicating the complete loss of hemoglobin by the red blood cells. Hypotonic solution is similar to the positive control, except that there is solute inside. The higher concentration of water in the solution, more than that in the red blood cells, so there is a net movement of water molecules from the solution into the red blood cells. Hemoglobin leaks into the surrounding fluid and turns the supernatant slightly red. Red precipitate is seen at the bottom of the test tube, indicating only some of the hemoglobin escaped into the solution and some of them still remain in the red blood cells. Since the concentration of water is less than that of the distilled water, there is no complete loss of hemoglobin from the erythrocytes. In hypertonic solution, the higher concentration of solute outside the cell causes water to move out of the cell down its concentration gradient. This is known as crenation. The supernatant remains colorless as hemoglobin is trapped within the cells, and red precipitate can be seen at the bottom of the test tube after being centrifuged. For isotonic solution, the amount of solute inside and outside the erythrocytes is the same, so there will be no net movement of water into and out of the cell, hence no change in the shape, size, and content of the red cells. The supernatant appears clear and red precipitate is seen at the bottom of the test tube. Section A2. From the graph plotted, it can be seen that the percentage of hemolysis is inversely proportional to the concentration of the sodium chloride solution. The higher the concentration of sodium chloride solution, the more hypertonic the solution and the lower percentage of hemolysis. The tonicity of the solution is compared and referred to experiment in section A1. Sodium chloride solutions of 0.1%, 0.2, 0.35%, they are behaving like the positive control, and in this section, as their percentages of the hemolysis are close to the one obtained for the control in section A1. The supernatant's dark red solution with white precipitates present at the bottom of the test tubes. The high percentage of hemolysis somehow indicates that solids in the solution is so few that it is almost negligible, causing net movement of water into the cells and allows almost complete hemolysis. Sodium chloride solutions with concentration 0.4, 0.45, and 0.5%, they are considered as hypotonic to the erythrocytes as they show close percentages of hemolysis and appearances to that of the hypotonic solutions in the section A1. The amount of solute in the solution is less than that in the red blood cell, creating an osmotic gradient between the erythrocytes and the sodium chloride solution. Water diffuses from the region of higher concentration to lower concentration into the cell, causing swelling of the erythrocytes and eventually hemolysis. After centrifuging, the cells that are seen as red precipitate at the test tube's bottom, with the supernatant as slightly red. Sodium chloride solutions of 0.55, 0.65, and 0.75% have the percentages of hemolysis close to that of the isotonic solution in section A1. So they are considered isotonic to the erythrocytes. Water move into and out of the cell at the same rate. They appear as clear solutions with red precipitates. The isotonic point, or the point of lysis, is determined based on the closest percentage of hemolysis to that of the reference isotonic position. Based on our elevation, we think that 0.55% of the sodium chloride concentration should be the point of lysis, with the 0.77% of hemolysis, although 0.75% of sodium chloride concentration has a percentage of hemolysis much closer to that of the isotonic solution, which is 0.71%. Probably due to the error in transferring the supernatant into the cuvette that we accidentally sucked some of the precipitate, giving us an inappropriate value. Sodium chloride solutions of 0.85 and above %, they are behaving as a hypertonic solutions. They have a similar appearances, which is a clear solution with red precipitate and percentages of hemolysis to that of the hypertonic solution in section A1. The high solute concentration of the surrounding solution draws water out of the red blood cells and causes crenation of the erythrocytes. Hemoglobin are trapped inside the red blood cells and they are appeared as red precipitate.
Section B. Tonicity is defined as the measure of the osmotic pressure of two solutions separated by a semi-permeable membrane. It is only affected by non-penetrating solids such as sodium chloride, whereas osmolarity measures the amount of particles in a solution regardless of the penetration property of the solute. It is assumed that the osmolarity of intracellular fluid of erythrocyte is 300 milliosmol. Sodium chloride, sucrose, and glucose are non-penetrating solids, which contributes to the tonicity of the solution. In the first 4 test tubes containing 300 milliosmol of sucrose, glucose, sodium chloride, and urea, they are theoretically isosmotic to erythrocyte. However, when compared to section A1, only 300 milliosmol of sodium chloride is behaving as isotonic solution to erythrocyte, whereas both 300 milliosmol of sucrose and glucose are neither isotonic or hypotonic if based on the value, or can be considered as a hypotonic if we really want to classify. This might be due to technical problem while carrying out the experiment, such as the precipitate at the bottom of the test tube is being disturbed during transferring of supernatant into a cuvette. For 300 milliosmol, 600 milliosmol, and 150 milliosmol of urea, the percentage of hemolysis are high. 150 milliosmol urea is hypoosmotic to erythrocyte, whereas 600 milliosmol urea is hypoosmotic to erythrocyte. It can be deduced that the urea is penetrating and has no effect on tonicity. The 300 milliosmol and 150 milliosmol of urea are hypotonic to the red blood cell, which subsequently cause influx of water into the cell and swelling of the cell. Hemoglobin is lost to the surrounding solution, giving the supernatant a red color. For 600 milliosmol urea, it seems that it behaves more like a positive control. In the test tube containing 300 milliosmol urea in 300 milliosmol sodium chloride, it behaves like an isotonic solution. Urea is penetrating solid, while sodium chloride is non-penetrating solid. It is hyperosmotic but isotonic to the erythrocyte. There is no net movement of water into or out of the cell, hence minimal hemolysis and the supernatant appears clear. Conclusion: percentage of hemolysis is inversely proportional to the concentration of sodium chloride for the range of sodium chloride 0 to 1%. For solution hypotonic to red blood cell, there will be influx of water into the cells and causes the cells to swell. On the other hand, solution that is hypotonic to the red blood cell causes shrinkage of cells as water moves out of the cell. The size of cell remains constant in isotonic solution where there are no net movement of water into or out of the cell. Thank you.